Now at the top of the hour, and we've started recording. So welcome everybody to the Diatom Web Academy. Thanks for joining us. You can see there in the chat window, we're having folks enter your name and location. It's just nice for us to know about our audience and get to see all the connections that we're making today. This webinar will be recorded and archived on diatoms.org. Our next webinar will be on April 28th on diatoms and paleolimnology, lake history through the lens of a frustral by Mark Edlund and David Burge. Details about the webinar will be sent out to the email list. And as Sarah said, everyone is muted, but please feel free to enter questions into the chat at any time during the webinar. We will monitor the chat for any clarifying questions for the speaker, and there will also be time at the end for questions. And with that, please introduce our speaker, Sarah.
Sure. Well, it's my pleasure to introduce Nick Schulte today. Nick did his master's degree at Florida International University, where he examined algal meta-communities in response to phosphorus gradients. Nick is currently working on a doctoral degree at University of Colorado. His research is directed at the integration of molecular methods into algal assessments. And so he has prepared a seminar for us today, which is really a great translation of what we can expect in terms of using Diatom DNA in environmental assessments. So, Nick Schulte, welcome.
Great. Thank you, Sarah, for the introduction, and thanks all for joining me today. My work is broadly, my doctorate is broadly on integrating algal DNA into environmental assessments. Today I'm going to speak specifically about using Diatom DNA for diatom-based environmental assessments. And I know that we have a broad, we likely have a broad spectrum of expertise in DNA, in diatom environmental assessments. So I'm going to be speaking at fairly high levels, but also to some technical aspects as well. I'll hope to clarify some terminology for you and mainly give you an idea of what are some of the techniques we can use and what's our general timeline to operation of using Diatom DNA in environmental assessments. I'm going to start with a couple of questions that I hope to address, that I will address in this presentation. The first of which are, what are the benefits of DNA methods in the context of environmental assessments? Secondly, how consistent or reliable are DNA methods for characterizing diatom communities? And then third is, how can Diatom DNA be applied to assessments in our current framework for how we do diatom environmental assessments, and also looking to harness the full power of DNA methods specifically. I want to go ahead and start right away with some takeaways that respond directly to those questions. As to what are the benefits of DNA methods, is that they're well developed as a general methodology and they're efficient. They provide reliable, informative count data, in this case for diatoms. They're cost, time, and expertise effective, and they can be easily updated and standardized with the proper coordination. So this aspect of using DNA methods is operational now. The methodologies are there. We just have to do a little bit more legwork at applying a consistent protocol, because there are methods, there are different decisions along the DNA processing pathway that can affect results. So we need coordination among programs, among researchers, analysts, and agencies to better reduce the variability in those methods decisions, and to be able to better achieve regular updates. And this is all readily achievable. Again, the DNA methods are there. We just need to put a little bit more effort into coordinating those methodologies among the people that are going to be using these data. And then thirdly, how can these methods be applied to bioassessments is that they're informative. So a lot of research has been done, particularly in Europe, especially the UK and in France, applying DNA data to their standard diatom assessments. And they show, and the results are generally that these data are informative, but they're not yet operational, especially for regions that haven't had that research put in to compare the DNA techniques to the traditional techniques of morphology. So the way that we can start to use DNA in assessments is to adapt DNA data to adapt existing biotic indices to DNA data, and this should be achievable fairly quickly. Or again, to harness the full power of the DNA data, we could design new biotic indices, and we're probably about 2 years away from being able to implement that in a variety of different regions. And again, needing region-specific studies prior to that to make sure that we know that these are working properly. So now I want to talk generally about some of the bioassessment challenges that DNA can potentially address. So what I just showed you were the main takeaways of this talk, and I'm going to kind of iterate and support those points as we go along. The first of which is, again, what are the benefits of DNA? So DNA can offer potential solutions to traditional bioassessment challenges, such as detection, in that rare taxa using morphology techniques are difficult to detect because of a relative abundance fixed count method that's very popular and widespread, and also that snapshot sampling can miss individuals. With DNA, we can detect trace amounts of individuals, and to alleviate that that deficiency of not being able to detect rare taxa. And also DNA can persist after an organism is gone, allowing us to integrate the environment a little bit more. And I guess you could see that as both a positive and a potential drawback, depending on the perspective and the interpretation of these data. So these are all questions that we have to respond to when we think about implementing DNA into bioassessments. The second challenge is accuracy, that just like some rare taxa are difficult to detect using morphological methods, some taxa are difficult to identify morphologically, especially those in ambiguous and cryptic species complexes. DNA offers potentially higher taxonomic resolution, getting to even intraspecific level resolution. And there's also a high potential and likelihood for consistency across analysts as long as we standardize methods that allow us to get to these DNA counts, or DNA diatom counts. And then thirdly is efficiency. Morphological IDs take time, up to 6 months per 100 samples, and money up to $1,000 per sample. And while DNA also takes time and money, DNA sequencing can take be up to 10 times faster and 10 times cheaper. And also they can be retroactively resequenced and updated cheaply and quickly as these methods become cheaper and become cheaper to operate and more updated. So I want to introduce you to a DNA technique called metabarcoding that I'm going to spend the bulk of my presentation discussing. It's a term many of you might have heard of that also goes by the terminology marker gene sequencing and amplicon sequencing. What happens in metabarcoding is that we determine community composition from short segments of DNA that target a biotic group, in this case diatoms. So those short segments of DNA are called barcodes, marker genes, or amplicons. So those are all synonymous terms. And what you're doing is you're looking at those segments of DNA for everything in the community that you sampled. What you get from metabarcoding is a table of taxon relative abundances in which taxa are identified either as morphospecies or given provisional names that are unique to the DNA sequence. Then once you have a count table with morphospecies names or provisional names, you can either assessment tools for you to use these DNA counts, or develop new assessment tools using DNA data. And this all costs from processing your samples all the way to getting your count data less than $85 per sample for most labs, and potentially cheaper, fairly much cheaper depending on the lab's capabilities. So in metabarcoding, there are about 6 steps to the process, and going from collecting the sample to extracting the DNA from that sample to sequencing the DNA that you just extracted, and then computationally converting those DNA sequences to count data, which is the field of bioinformatics, then naming those processed sequences using morphospecies taxonomy or provisional names, and then applying all of that to the assessment framework. So what I want to do today is to go step by step a little bit to show you the reliability of each point in this process. And there are a lot of decisions that are made within each of these steps that I do not have time to get to today. So please reach out to me if you have any questions. We can discuss these after I finish speaking. But for now, I'm going to address one clear question within each step that represents some of the decisions that we have to make and how those decisions could affect the assessment. So we're just asking, how well developed are these methods, and how do the different decisions we make in these methods affect our final assessment? So for sampling, I want to ask the question, are there differences between a biofilm and water sample? So traditionally with morphology, we go to an aquatic ecosystem, and for the most part, I'm going to be talking about wadable streams and rivers, but much of this can also be applied to lakes and wetlands, given the appropriate adaptation. But say we go to a stream, our traditional methods is we sample a biofilm, collect the biofilm, process the biofilm for diatoms, and look at those under the microscope, and then use those to determine our ecosystem assessment designation. One type of DNA sample, which is very similar to our traditional method, is called bulk DNA, which is DNA that comes directly from the organisms. So in this case, when we're targeting a biofilm, we're targeting diatoms, and we would consider that a bulk DNA sample. So in which case, the sampling process is the same as with morphology, and what we currently do is that we go out to a stream, collect a biofilm, but instead of digesting off all the organic matter and looking at it under a microscope, we extract the DNA and process that through the rest of the metabarcoding processing framework. The other type of DNA that many of you have probably heard of is environmental DNA, or eDNA. This type of DNA is commonly used to refer to all types of DNA collected from an environment, including from a biofilm, but that's not technically correct. eDNA is DNA that's shed from organisms, that's extracellular from organisms, and that you're specifically targeting the DNA that's extracellular. So a biofilm might contain some extracellular DNA, but the main goal there is to sample the diatoms. For eDNA, instead, your main goal is to target the shed DNA, the extracellular DNA that's suspended in the water column, in which case you go collect a water sample, filter the water, and then extract the DNA and go through. For the most part, bulk DNA resembles our current method more so than eDNA. But let's see how these compare compositionally. So bulk DNA versus eDNA in recovering taxa, or in the number of taxa. So in this one study in stream biofilms in New Zealand, they found that bulk DNA and eDNA from the same streams on average had 29 of the same taxa, but bulk DNA only had 1 unique taxon, and eDNA had 58 unique taxa, showing that eDNA is integrating DNA and individuals from across the environment, whereas bulk DNA is more restricted to that biofilm alone. So these two methods are showing us different compositional, differences in composition of the communities, and likely will affect our final assessment. A lot of work hasn't been done on actually applying eDNA to assessments in diatoms. Most of it has been on bulk DNA. The main story here is that the main takeaway is that bulk DNA, eDNA tell different stories. Bulk DNA is most similar to current methods, and it may be more difficult to interpret eDNA because it is integrative, because you have to consider things like hydrologic flow paths and other potential sources of that DNA. So for now, my sampling recommendation is for the most immediate implementation of DNA into bioassessments is to use bulk DNA for our standard assessments. I also want to say that even if you are not currently using DNA in your program, it is worthwhile to take and store DNA subsamples for later analysis, because the field is moving in this direction, and we don't know the extent to which it will move in this direction, but especially if you're looking at long-term records, it makes sense to collect these samples now for very cheap, store them for very cheap, and you can always process them later down the road. So overall, DNA sampling methods are fairly well established, and they're operational now. We just need to standardize a little bit, and more research, if we want to get to that that eDNA spot, needs to go forward before we implement eDNA in our regular sampling protocols. The next step in the process is DNA extraction, so extracting the DNA from that bulk sample you just took. And the main question here is whether extraction methods affect composition. There are several different ways of extracting DNA, from using commercial extraction kits that provide you reagents, provide you all the supplies you need, as well as step-by-step instructions that are really easy to follow. There are non-kit extractions, which are similarly reagent-based, but they tend to be cheaper than the commercial kits. And then there are instrument extractions, which use both commercial and commercial kit and non-kit extraction methods for higher throughput. So to make the time of extracting DNA about 25% of what it would be on the benchtop. But the main question is whether these extraction methods differ for our final assessment or our final community composition. And the main answer is they don't really. So here on the y-axis is a biotic index score for which 8 different environmental samples for stream biofilms were run. And each bar for each sample is a different DNA extraction method. And you can see that there's not a huge difference in the final biotic index score, so that the extraction method really isn't driving the differences in assessment between different samples. So at this point, assessments are more or less consistent among DNA extraction kits. And I recommend for those that are wanting to implement this now and very easily, who might not have a lot of existing infrastructure to run DNA, is the QIAGEN DNeasy Power Soil Kit is the most straightforward and has quality, reliable results. But the Sigma Aldrich non-kit DNA precipitation method has been recommended for standardization by some research groups on diatom bioassessment in Europe, and it is cheaper. So that is a way that we can move forward and, again, work towards standardizing these methods. Excuse me. So DNA extraction, operational now. We just need to kind of work out the choices involved on who's using what, and thinking about that as we go forward. So once we have the extracted DNA, we need to sequence that DNA. And there are several different aspects of sequencing DNA, but I'm going to focus on what gene regions should be targeted when we're sequencing the DNA, because again, with metabarcoding, we're focusing on specific short segments of DNA that are supposed to be taxonomically informative for the group that we're interested in this case, diatom. So we want to focus on, of all the genes in a diatom genome, we need to focus on a specific one or a specific segment that allows us to identify species across the board. So in DNA sequencing, there are 5 major steps. The first is to select a pair of primers that target that short segment of DNA that we're interested in. And a primer is basically just a pair of sequences that border the target sequence. They latch on to the DNA and basically say, hey, we need to sequence what's in between us so that we can get that taxonomic information. Then once those primers are attached, we amplify using a technique called polymerase chain reaction. We amplify that short segment of DNA that's in between those two primers. And that short segment of DNA, again, is called an amplicon or a barcode or a marker gene. Then what we do is we attach a unique sample ID to each of those amplicons, each of those copies of short segments of DNA, and that sample ID is just in the genetic code language, so A to C, T's, and G's. Then what we do is once everything has a sample ID, all the DNA has a sample ID associated with it, we put it all together from all the different samples we have, and we sequence all of that on some sequencing platform. So there are three major places where we have to make some decisions. Number one is what primers do we use? Number two is how well do we amplify, or what process do we use to amplify? And third is what sequencing platform to use. Amplification and sequencing are pretty standard across the board, but primers are more specific to the group of interest. So we want to focus on what primers are good for diatoms. And some work by Sarah Hampshire and colleagues looked at some morphospecies that were identified and extracted DNA from, and looked at 3 different primer pairs that targeted 3 different amplicons to see how well they recovered those morphospecies. And this 23S, these long black bars, show that it's identifying that there are taxa there, but they're merging multiple morphospecies together. So 23S is merging Nitzschia brevirostris with Nitzschia pusilla and Nitzschia laevis. So it's not taxon species specific. This COI has a lot of white blank spots, which means that it didn't even see those morphospecies in there at all. But this RBCL is kind of that Goldilocks in the middle, where there are shorter black bars, which means that it's recovering those species-specific taxa morphospecies, more or less, at least better than these other 2. So primer selection determines the phylogenetic or taxonomic resolution with which we can identify diatoms using DNA. And some primers have been developed that target diatoms, and they're good at distinguishing diatoms at the species level and even subspecies level. Specifically the RBCL gene, which is located in the chloroplast, and the 18S V4 region, which is found in the nucleus. There are also other primers that are called universal primers, and these are primers that target broad groups like all eukaryotes, all plants. They're supposed to be a silver bullet, but they're not for the level that we need diatom identifications at. At best, they're good for some species, but usually just genus, reliably genus. So they're not recommended for diatom bioassessment. We really should be using these diatom-specific primers, and I recommend using this one pair that was developed by Martin Kelly and colleagues in the United Kingdom that targets an RBCL region that's good for diatom species resolution. So DNA sequencing, the methods are all there. We just need to kind of fall in line behind one primer set that we can all use, so that all of our agencies are consistently using the same identification tool. It's basically saying we're using the same book. It would be analogous to saying that we're using the same reference book for our morphological identifications. So now that we have our DNA sequences, we need to process those sequences computationally, which is the bioinformatics. So we want to ask, the main question here, there are several, such as how do we get rid of bad quality DNA sequences? What's the software that we use to process these sequences? Those are relevant questions, but there's a fair amount that's being done on that. I want to focus on, should sequences be grouped together? And basically, you can group unique sequences together by percent sequence similarity into taxonomic units called operational taxonomic units, or OTUs. This is a term many of you are maybe familiar with. And basically what you're doing is you have 2 unique sequences, but these sequences are, let's say, 99% the same. So we just merge the 2 together and call them the same taxon. It used to be that 97% sequence similarity was seen as species level resolution, but more and more research is suggesting that we do not group unique sequences together and that we keep unique sequences separate into exact sequence variants, or ESVs. These are also called amplicon sequence variants, ASVs, and zero radius OTUs, ZOTUs. I'll be using ESVs throughout this talk, but just know there's some jargony terminology that we also have to agree upon to make this as simple and easy to understand for everybody. So what does grouping do for assessment? For OTUs, the grouped sequences together, this one research group looked at assessments using OTUs versus assessments using ESVs relative to an environmental gradient, and found that there was a stronger correlation between ESV assessments and the environmental gradient than the OTU assessments, suggesting that ESVs outperform OTUs in assessment in river biofilm diatoms. I recommend using ESVs going forward because this, again, maximizes the amount of information that we can get from DNA rather than merging information together that could be potentially useful. One thing to bear in mind is that bioinformatics can be easily updated or changed in the future. We can always go back, and let's say 3 years from now, we were all using a certain quality threshold for filtering out bad reads. We can always go back and change those quality thresholds very quickly, very easily, and very cheaply. So that is a big positive for working towards updating methods to the best possible that we can bioinformatically. So again, bioinformatics are ready to go for the most part. We just need to rally behind a couple of specific methods decisions. Then once you have your grouped or ungrouped ESVs or OTUs, switch those there, apologies, into a count table, we need to assign those ESVs some taxonomy, some name, whether that's a morphospecies name or just a provisional name that's specific to that DNA sequence. And then we have to think, if we're assigning morphospecies names, is that taxonomy reliable? So there are 3 ways we can assign taxonomy. We can assign ESVs to morphospecies, which I call reference-based metabarcoding. This is done by percent similarity to a reference sequence. So they're somewhere out there, somebody has cultured some diatom that they know 100% is that diatom morphologically. And then they've sequenced that diatom for the amplicon of our interest. Then when we have our environmental sample, we can have our own sequence and see if it matches up with that sequenced named reference diatom. You can do that for all of the different ESVs you have in your sample, and you remove all unnamed ESVs and potentially aggregate all of the ESVs into the same names. And this allows you to retain morphospecies ecological information. However, ESV names are only as good as your amplicon resolution, which we already talked about, and your reference library completeness. The reference library is just the database of sequences identified by morphospecies with reliable morphospecies names. The biggest thing here is that we need more morphospecies verified reference sequences because there are very few curated diatom reference sequences relative to described morphospecies. The best database for diatom sequences is something called DiatBarcode, which is very good for RBCL in particular, but it's mainly used for European taxa so far. There are also non-curated databases like NCBI's GenBank and Silva, but these morphospecies names are going to be a lot more suspect than this DiatBarcodes. The ways that we can expand these reference libraries, number one is just putting funding behind it, so that we can expand via culturing, single cell genomics, and then even looking at taxon or environmental samples that are dominated by one taxon. Another way of dealing with taxonomy for DNA sequences is to not assign ESVs to morphospecies And this is this called taxonomy-free or taxonomy-blind metabarcoding. Here, we just use provisional names that have no morphospecies ecological information. But the plus side is that you keep all of the high-quality ESVs in your sample. And then you could do kind of a mixed version of this, where you attach names, morphospecies names, to whatever you get, but you don't delete ESVs that are unnamed. You have to use these morphospecies either in a reference-based assessment way or just kind of an ecologically informed way. That's not going to be a standard assessment tool, probably. Just comparing reference-based to taxonomy-free metabarcoding, we can see here that the light gray is taxonomy-free versus the darker gray, which is reference-based, that for the proportion of sequences that can be used in the biotic index, as well as the proportion of OTUs, taxonomy-free uses most of the data across the board, whereas reference-based does not. There's variability in this one sample, for instance. All of the sequences were able to be attached to reference names, whereas in this one, almost none of the sequences were able to be attached to reference names. So taxonomy-free sequence counts and OTU numbers are higher than reference-based. And you can imagine that that could potentially affect our final assessments. For now, I'm not going to recommend either reference-based or taxonomy-free because I think there's positives to both methods. I would say that reference-based is the most easily implemented into our current assessment infrastructure. But regardless, we need to expand reference sequence libraries to make sure reference-based metabarcoding is as optimal as possible. We also need to be conservative with our percent similarity matching for reference-based. We want to make sure that we're not assigning names incorrectly to DNA species. And if possible, avoid discarding ESVs that are unnamed unless it's necessary for reference-based assessments. Just like with bioinformatics, taxonomy can be easily updated or changed in the future, and really will be constantly updated as reference libraries expand. So taxonomy, there are those reference-based and taxonomy-free methods that are good. Solid reference-based is probably the more directly applicable to assessment. So what does all of this mean for our final assessment? We saw that there's variability within each of these processing methods, but how well can diatoms be used, Diatom DNA be used in environmental assessments, and how does it compare to morphology? So one way that we can use Diatom DNA in assessments is that we put them in existing biotic indices as is. This only works for reference-based metabarcoding, but given that there are ambiguous identifications with DNA, and also that there are very few reference sequences, means that these biotic indices were created with a certain size species pool expected based on morphology, but that species pool is going to be a lot smaller for reference-based DNA because we don't have as many sequences. So if you are going to use existing biotic indices with DNA data right now with no changes, I would strongly urge you to interpret that with caution and compare it to morphology. And really, we shouldn't be doing it. We should be adapting these existing biotic indices to DNA data. And that means recalibrating the biotic indices, rerunning the models that underpin the indices and the ecological values that underpin the indices, and improve the reference library so that the species pools between the morphology and the DNA are as similar as possible. Third is that we can develop new biotic indices for DNA data, which should be able to allow us to harness as much information as DNA will allow us to give us. And this can be done for both reference-based and taxonomy-free metabarcoding, but it requires additional reference site sampling, index design, de novo ecological values for provisionally named taxonomy-free sequences. But it allows us to look at environmental relationships with DNA data that might not exist for morphospecies data. For all three of these methods, there's still a need to compare DNA-based assessments to morphology-based assessments. we are not at the point where we can just get rid of morphology and replace it with DNA, because there's still a lot to understand about how the two complement each other, where there's novelty, where there's redundancy. And this is because there's differences in morphological and genetic variability. Here in this A, for morphology, we have two different taxa, this one here and this one here. But genetically, those two taxa have a difference of 20 to 1 DNA molecule. And I want to make sure I emphasize, I mentioned it, but I didn't emphasize it earlier, that metabarcoding only gives us relative abundance. We cannot get absolute abundance from DNA metabarcoding. So here we would count this diatom 20 times and this diatom 1 time, whereas morphologically we would count each of them only once. You can also have species that are morphologically close, and in morphology counts you would call them the same thing, but genetically you would call them different things because there's cryptic genetic diversity there. You can also have dead cells that are in morphology that you count as part of your assessment, but no DNA for those dead cells. Likewise, you can have extracellular DNA that's not associated with a diatom frustule in morphology. So there are morphological and genetic differences in what we're measuring, and that can have that in turn affects the species composition as well as the final assessment that we measure. So when we compare morphology to reference-based metabarcoding as far as the species composition returned, we find that 13%, in this one study found that 13% of the taxa were shared, but that there were 173 unique taxa to morphology and 35 unique taxa to DNA. And this was based on OTU grouping. But you can see this 142 here is the number of morpho species that were not present in the reference database. So there was no way you could have recovered those 142 taxa in reference-based metabarcoding. So metabarcoding community composition and diversity differ from morphology, but what about assessments? In some work on diatom streams in Europe, they compared reference-based metabarcoding biotic index scores using an adapted index to morphology biotic index scores and saw a fairly strong relationship, an R-squared of 0.57. So a strong relationship, but there's definite variability. Here, the morphology biotic index score is 2, whereas the metabarcoding one is 5. And they see the same sort of pattern with taxonomy-free metabarcoding assessments to morphology assessments, if not even a stronger relationship. So there's stuff that we need to tease out there, but generally reference-based and taxonomy-free metabarcoding seem to be consistent with morphology, but not the same as morphology. So those were both adapted indices. What about newly designed indices that are meant for metabarcoding data only? We see that there's a similar relationship between morphology and the environment and reference-based de novo assessment and the environment, suggesting that de novo metabarcoding biotic indices, so newly developed biotic indices, are as associated with environmental impact as morphology. So my recommendations for assessment are in the short term that we should be adapting existing biotic indices to reference-based metabarcoding counts. This will be as similar to what we currently do morphologically as possible and allow that interpretation gap to be minimized while still allowing us to use DNA and morphology data complementarily. In the long term, we should look into developing new biotic indices for both reference-based and taxonomy-free counts to better harness the power, the untapped power of DNA data. Assessment is really the one place that we're not operational now. From sampling to taxonomy, we have a good handle on the methods. Assessment requires a little bit more information, a little bit more research before we're ready to go whole hog in on DNA by itself. It doesn't mean that we shouldn't be collecting DNA up to that point and implementing it into our assessments. It just means that we need to use those data with caution in assessments right now. So returning to our original question of what are the benefits of DNA methods? how consistent are DNA methods and how can DNA be applied to assessments? I want to contextualize these responses in kind of a timeline or an operation context. So the benefits of DNA are that they're cost effective, they're efficient, and that processing from sampling through taxonomy is well developed and operational now. However, it requires standardization because there is variation in some of those methods. So anybody intending on implementing DNA into their assessment should be willing to work towards standardized and updated methods so that we do not repeat the mistakes made in morphology in terms of inter-analyst and inter-program inconsistency. This is all readily achievable, and there are groups that are working to accomplish this, and you can join in. I'm sure they would be happy to welcome you in. And thirdly, how can we apply Diatom DNA data into assessments? Since assessments are region dependent and we need research at that scale, they're really not ready for immediate implementation. If you want to use metabarcoding data for to look at environmental relationships, that's operational now. Retrofitting existing biotic indices for reference-based counts is readily achievable and can be done fairly quickly. De novo biotic indices are probably 2-ish years away for some regions, and I would say that folks in the EU have been working on this more intensively than here in the United States. So those will likely be fully operational before the USA and other regions, but we can work with those research groups and agencies to learn from what they've done and to coordinate with them. Really quickly, I know many of you are probably wondering, what about morphology? I said that the methods complement each other, but they don't replace each other. More research is needed to determine the redundancy and novelty of each method. And for now, we need to continue to collect and use morphology and assessments until we understand the relationship between the two of those. There could be a point in the future where metabarcoding replaces morphology for assessments, but we're not there yet. And it's possible that we won't be there. It depends on the information that we're interested in and how much we trust each data source. Regardless, we need morphologists now and in the future for building those reference libraries. We need confirmed high quality taxon identifications associated with sequences. So that is a clear place where morphologists can work in addition to their current work in morphology assessments. I talked about metabarcoding throughout this talk, but I want to mention 2 other DNA-based tools that could be useful in assessments very briefly. The first is qPCR, or quantitative PCR, in which we measure gene expression of a single taxon-specific gene to get the absolute abundance of that gene in a sample. This is best for single taxa of interest, like nuisance and invasive taxa, potentially like Didymosphaenia geminata, where you're interested in the absolute abundance of it across different samples. And this is pretty cheap at less than $50 a sample, but there aren't a lot of taxa that we are currently able to do that for in terms of diatoms. The second is called metagenomics, which is also called shotgun sequencing. This is similar, I would say, to how we use environmental DNA. We use it as a broad term to encapsulate anything we're doing with environmental DNA, environmentally sourced DNA, but metagenomics is really a specific method that's kind of like metabarcoding on steroids, where metabarcoding looks at a single gene segment that's highly variable for a specific group so that we can identify that group to a species. Metagenomics looks at every gene for every individual in a community sample. As a result, you get genes that are both important for taxonomy and for function. And you also get non-diatom individuals that you sequence for. You get ducks, you get humans, you get bacteria. As a result, you might get too much information for what you want if you're just focused on diatoms at this point, and it's a little bit more cost prohibitive. However, sequencing costs continue to decline, so metagenomics could become more efficient than metabarcoding, but that does not mean that metabarcoding will become obsolete. Rather, you will be able to use metagenomic data to focus in on that one gene region that you used for metabarcoding and basically use metagenomic data like you use metabarcoding. So it would be consistent with previous metabarcoding data. So there's no concern that you should wait on metabarcoding for metagenomics, because you can always do the metabarcoding processing for metagenomic data. Like I said, there are efforts underway to coordinate all of this and to increase our ability to understand diatom-based assessments using DNA, including training opportunities, pilot studies, stakeholder workshops, and key collaboration among researchers and managers. So please look into these. I've provided in presentation references. There will be a reference page posted to Diatoms of the United States, and I'll try to I'll annotate that as well to better position you to look into this stuff more. So I'll leave you with some words of the late great John Prine, that we are living in the future, and with diatom bioassessments, it's time that we use diatom DNA to recognize our future capabilities. That's it.
Thank you, Nick. Thank you, everyone. We have a few questions in the chat that I can start with, and if you have questions that you'd like to have Nick address, please put them in the chat window, and I will go ahead and read them so they'll be included on the recording. So Nick, how much material do you recommend that people collect and store for later analysis, and how do you store that material?
Great question. I had hoped to discuss this in more detail, but I had to trim it down. As you can see, I'm long-winded. So currently, I think it's sufficient for a biofilm to collect 2 milliliters of homogenized biofilm composite sample. You can use the same composite sample that you currently use for morphology. And I recommend storing it in the short term, if you're going to extract the DNA fairly quickly, at -20 degrees Celsius, so freeze it. And the long term, at -80 degrees Celsius, it should last for several years, if not much longer, before extraction. And after extraction, you can do the same thing, freeze at -20, -80, and it will last years.
Okay. Do the index comparisons that you showed include morphology and bulk DNA analysis and not eDNA? I'm not sure.
Yes, that is correct. Nothing I showed you was eDNA except for that one slide Venn diagram comparing bulk and eDNA taxon richness. Everything else has, pretty much all the research on diatom assessments using DNA has been done with bulk DNA, so biofilm samples.
Okay. Thank you for the talk. Is anyone using Diatom DNA in paleo studies yet?
They are. Next Diatom Web Academy is on paleolimnology, and I'm hoping that Mark Edlund and David Burge have a little bit of more information than I can give you about using Diatom DNA in paleo studies, but it is being done, and it is being, I think that there is recovery of diatom sequences. Don't quote me on that right now, but there have been several studies for other algal groups and diatoms for ancient DNA that we can do it. The methods can be adjusted a little bit because you're dealing with degraded DNA a lot of times, so you can do different steps in that processing to increase the detection.
Okay. Anybody else have questions? I'll go ahead and insert one here. You talked about diatoms here. What is the potential for using other organisms in bioassessment that have not been currently used? For example, things like sponges or bacteria or different fungi that could be recovered with DNA.
Yeah, I think there's a strong potential for it. There is a recent paper out from Alison Minorovich and Marina Potapova and colleagues that looked at one of these universal primers for eukaryotes. Then they focused in on diatom sequences from there, but also other eukaryotic sequences, and found that there are strong environmental relationships with other biotic groups to environmental impact, and that those relationships are different across those biotic groups. So diatoms showed different responses from ciliates, for example. So so DNA opens up a new group of organisms for us to look at, especially bacteria and archaea, that assessments really haven't focused on outside of looking at specific taxa for like Escherichia coli for impact.
Okay. Stefano says, thank you, Nick. Have you heard about some relationship between diatom DNA analysis and abnormal valves? For example, teratological forms, about some mutations that are potentially DNA based that leads to these abnormal valve formations?
I have not. I hope that work is out there, and I feel like there has been at least one study that looked at acid mine drainage diatom DNA, so that would be an interesting study to look into. But I'd say there's a high potential for that. Whether those mutations would show in the gene regions that we target for our current identifications is an open question, however. There might be mutations elsewhere in the genome, but they might not allow us to identify teratologically mutated taxa using our normal metabarcoding methods.
Flawless presentations, great slides, thank you. What would you recommend for an already ongoing monitoring program that uses morphological data? Should we start switching to DNA or keep only morphology? I assume that means like if you can't do both.
Yeah, I think that's the biggest question, is when you can't do both because resources are limited. I would say at this point, morphology is important to continue. And there likely needs to be some overlap, however difficult that might be, just to make sure that if you want to make the transition to DNA, that you're not putting all your eggs in one basket, and then you can't use your past assessments anymore. So what my recommendation is to continue using morphological data for now, but collect DNA now. Store it at -20, put it in the freezer. You don't have to, if you have access to a -80 somewhere, that's great too. That's even better. But maybe wait for a little bit more work to be done in your region, especially before you go all in on DNA.
Okay. Regarding extraction of DNA, are some diatoms more resistant to others than to cell lysis?
I don't think that has been looked at specifically, but there is an expectation that highly silicified diatoms might be more difficult to break apart. However, in most extraction methods, there's either a physical or chemical process that's forcing these frustules open. And so far, based on the results, it doesn't seem like there's any taxon-specific, like any glaring holes in taxa that we're unable to recover using those processing. So I would say that for the most part, that's not a major issue.
Okay. Is the concern with eDNA in streams that there would be soil diatom DNA coming into the stream during rain events and other periods of greater hydrologic connectivity?
I can imagine that could be one concern. I think the greater concern is more of an interpretation, in that we're used to having composite samples from, let's say, 10 or 11 transects that are snapshot samplings of a biofilm that's growing on a rock right then and there. eDNA could potentially integrate diatom DNA from who we don't even know how far upstream. We need more work done to know what we're sampling with eDNA. And that's the main question, is we don't have as much of a handle on what eDNA is measuring as we do for as we do for bulk DNA. If we just cared about taxon presence in any stream, regardless of reach, the entire upstream region from your reach, then eDNA could potentially be fine. But there's still a bit of that interpretability problem. You could also potentially have issues with degraded DNA that you don't have as much of a problem with bulk DNA.
Okay, great talk. Do you think a cell-free approach using functional gene abundance rather than species abundance might gain popularity to avoid the need for reference databases?
Yeah, I can imagine that being informative and speaks to the power that metagenomics could unlock, or metabarcoding for a functional gene, that we could potentially generate new biotic indices based on functional gene relative abundance. The problem I can envision is that it would be relative abundance data for the most part, unless I guess you could use qPCR as well. In short, I think it could become popular, or at least another tool in our tool belt. But I also want to emphasize that there is a cell approach that doesn't use reference databases at all, and that's the taxonomy-free approach I was talking about. There haven't been a lot of assessments, research on assessments with those, but I think there's a great potential there for creating new data or new biotic indices using taxonomy-free, raw, unique sequence data, and specifically looking into things like O over E models, observed over expected models, which basically look at species loss. And what you could do there is just look at sequence loss. ESV1, has it been lost or gained in this stream? And you could look at something like that without having the need for reference databases.
Great job, Nick. Beyond reference databases, the amount of species identified between genomics and morphology is outstanding. Why do you think that is? Is there a problem with clustering OTUs at 97%? Or perhaps the morphology species assessments are using features that are more prone to an overassessment of variability?
I love this question because it speaks to some of the things we don't know yet about how morphology and DNA sequencing differ. I would say definitely that clustering OTUs at 97% is arbitrary, and we do not have a firm handle on, for different amplicons, what that species resolution actually is. Are we getting species level resolution for all taxa? Are we doing it for a subset of taxa that have a higher variability? For instance, maybe all Hanschia genera are at species level, but all Navicula taxa are only being identified at genus level or something. I don't know if it's that severe, but there's a possibility that that clustering muddies the waters a little bit. I would also say that based on talks with Bart van de Wever at the Belgian Botanical Garden, is that they're finding that they've seen that there are some taxa that he can identify he can differentiate morphologically that genetics cannot differentiate, and vice versa, that genetics can differentiate a taxon that looks the same to a morphologist. So there's definitely some cryptic diversity on both sides that are probably lending themselves to those differences in species capture.
Okay, another question. Tom uses eDNA to look at nutrient effects in Nebraska streams, and he likes how they are potentially both relevant at basin level scales as opposed to more localized biofilm samples. So that's a comment.
Yeah, I think that's a great point. And one of when on that bioassessment challenges potential DNA solution slide I listed under potential DNA solutions that that DNA integrates the environment and persists over time. And that can be seen as a value. And I think for eDNA, it is a value, and it's something that we want to harness. But it can also be a challenge in and of itself for interpretability, because we don't have as firm a handle on basin level scale diatom dynamics as we do at the local scale, just because of the history of research that's been done on diatoms in environmental assessment. But I agree, eDNA is a very powerful tool. It also has the potential benefit of being able to use a single sample for multiple biotic groups, for fish, for inverts, and for diatoms. It's something that's very alluring about being able to just go fill 3 liters of water, filter it, and then that's the sampling you need to do at the stream, right? Like, that's great, and it would be great if we could get there. My advice right now is that we are not yet there, and we need more research on there.
Okay, last question from Zavi. Can we infer biogeographic patterns using DNA data? And if so, how would you compare this to morphologically based studies?
Yes, I think that we can definitely look into biogeographic patterns, and because we can have consistency in sequence identification across large spatial and temporal scales, we can see whether the same sequence that we see in a Tennessee stream is present in California or in Europe, and how much variability there is. So there's a huge potential for both community level biogeographic community analysis, as well as population and metapopulation biogeographic analysis.
Okay, well, we're back at the top of the hour. And so I'd like to thank you, Nick, for an excellent presentation. There are a lot of great words coming through on the chat, and appreciation of your talk and how clear it was, and really being directed at the things that people want to know. So thank you. And I'd like to thank all of you for joining us here today. It's been really wonderful to see all the names out there of people that I miss, and please take good care of yourselves, and take care of your families and your neighbors, and we will learn new things and come out of this disaster better. So if you did not get a chance to ask a question, or you think of a question later, please contact us at the Taxonomic Certification Committee. You can access that on the Diatoms of North America page, or anywhere else. I hope. We recorded this presentation, and so it will be posted. It usually takes us a couple of days to get it posted. And please join us 2 weeks from today for a presentation on diatom paleoecology. So thank you, everyone. Really appreciate you being here.
Thank you all.
Till next time. Bye.